Okay, so we're going to move into endocrine. So this is chapter 17. Again, all these questions in the PowerPoints, you guys should be reviewing those as you're studying outside of class after this lecture so that you can answer all those questions satisfactorily before the exam, since the questions on the exam will be pulled from these review questions. This semester, we're going to have fewer questions on the exams because most of my classes can't double up on time. So you're only going to have your hour and 15 minutes. It's only going to be 50 questions. So each question is going to be worth more. So it's going to be harder to get a good score if you missed a huge chunk of material. I'm sorry. I can't help it. There are classes scheduled before and after. I can't help it. So be sure you know what you know before you go into the exam. Yes?
Are you going to be posting a review to like other classes?
Of course. You mean like a list overview? No, that's these questions.
Oh.
They're posted in the PowerPoint. That's it, right there. Those things.
So for the exam, they should just do all these questions. And
Yep.
Then,
That's where I'm taking all of the questions from.
Okay.
They will be questions that meet these points. All these reviews, all these objectives. If you are clear on all of those things, you will be fine for the exam. Cool? Deep breath. It's okay. All right, so we're going to move into endocrine system. What is the endocrine system? Skin? What? Huh? Hormones. Hormones, yes. Hormones are released from glands. All right, so what is a hormone? What's specific about it? They're weird. Okay. How? How are they weird? How are they different from other signaling molecules? They travel in the bloodstream, exactly. So they're released in one place, and they travel through the bloodstream to get to their receptor. The receptor is distal to the site of release. The receptor is far away from the site of release. So the target cells for each hormone have receptors specific for that hormone. They bind that hormone once it's released into the bloodstream, and then they do something in response to the presence or absence of that hormone. So a hormone is secreted from a gland into the interstitial fluid around that gland. It then travels into the bloodstream, through the bloodstream to the target tissue, and then goes into the target tissue, into the interstitial fluid, binds its receptor eventually, causing some action. So it's very similar to the nervous system with respect to the idea that each of these molecules being released, these signaling molecules, are ligands. They are things that have to bind something else. And when they bind something else, they bind their receptor, something happens. The only difference is the spatial limitation of a signaling molecule within the nervous system versus one through the endocrine system. Endocrine system can signal across large distances. The nervous system generally does not. When a neurotransmitter is released from a cell, a nerve, a neuron, it acts right there on the postsynaptic cell. When an endocrine gland cell releases its hormone, it acts typically very far away from that origination point. The nervous system causes pretty specific effects. You have to recruit lots of different neurons and make them signal to lots of different areas if you want a widespread effect. The endocrine system can have widespread effects because these receptors for a specific hormone can be located at multiple places through the body. So because this is traveling through the blood, as soon as it hits a tissue that has that receptor, it can then act on that tissue. So you can have widespread effects far faster with the endocrine system relative to the nervous system. Also, it lasts longer. We can have long downstream effects from the endocrine system. The nervous system is just sploosh, neurotransmitter released, it binds, it does something, and then it's taken back up again. Endocrine system can trigger downstream effects like changing gene transcription, changing signaling cascades, up or down regulating receptors, making new proteins, breaking down proteins, signaling to other endocrine cells to release different hormones, or, pushing babies out of a uterus. things like that. That's a long-term effect. Yeah, that's like an 18-year-long effect. So the difference with an endocrine gland is, what's that? Or longer. It depends, like 26 now, right? The IRS lets you stay. It'll keep counting you. Okay, so an endocrine gland cell produces a hormone. It releases it into the interstitial fluid and then travels into the bloodstream. It'll go through the bloodstream to its effector cell. Ooh, come on. Oh no. RAM. Need more RAM. Okay. So once it gets to its effector cell or its target cell, it exits the bloodstream and binds to a receptor there. And then that target cell does something. Depending on the hormone, it'll be cool or really, really cool. A neuron, a little different. Or if you remember from last semester, a nervous system signal comes down an axon to a target cell. We have a synapse onto that target cell, and something happens specifically within that target cell. Neurotransmitter is released, it activates a receptor, and then some downstream thing happens inside that cell. But that's as far as it goes. This is more complicated. If you notice, all the material is progressively getting more complicated. So there are 4 primary functions of the the endocrine system. First of all, it regulates growth and development. So that's the initial function of our endocrine system, is to make sure that our metabolism is kept exactly where we need it, just high enough so that we're keeping enough energy coming into our tissues to make them function properly, but not so high that we're depleting our energy stores. It regulates development and growth of embryos, so development of gametes as well. And it helps to move along the process of tissue differentiation and embryonic development. It also helps to maintain our blood volume and blood composition. So primarily electrolytes are mediated by hormones through your kidneys. Glucose as well. So blood glucose levels and electrolyte levels are 2 big targets of hormone regulation in the body, and those are 2 things that we can manipulate clinically by either putting in hormones or hormone antagonists. So those 2 things help regulate blood volume. So blood volume is indirectly regulated by hormones. Digestive processes are also regulated by hormones, and reproductive activities are regulated by hormones. So gametes mature in response to hormones. Secondary sex characteristics develop in response to hormones. Here are your questions. Again, review these questions. So endocrine glands are where all the action is going to take place as far as getting these hormones ready to go. So these are epithelial tissues typically that have a connective tissue matrix or framework around them. Some of these glands are endocrine organs, and some of these are other organs that have an endocrine function. So like the pituitary gland is a strictly endocrine organ. The adrenal gland, the thyroid gland, strictly endocrine organs. But your skin, Caleb said skin earlier, and I don't think you meant to say skin at all, did you? Yo, you totally did though, right? Yes, definitely meant that as an answer, because skin, although not primarily an endocrine organ, has endocrine functions. Just like your heart, your liver, your stomach, adipose tissue, your intestines, your pancreas, which is primarily for digestion, but produces and responds to hormones. Essentially, you are a walking endocrine machine. Everything you do, okay, all right. There's no such thing as love. Everything you do is because of hormones. They're just chemicals. Every single thing you do every day, day in and day out, it's just chemicals. Sorry. If you start crying right now, it's because of hormones. And it's because of these specific hormones, these specific chemicals control most of our emotions, most of our actions. Every decision we make is in some form or fashion influenced by hormones. So here are your major endocrine organs. So over here, we've got a lot of them. Thyroid, pineal, pituitary, parathyroid, and adrenal glands. But over here, there are more tissues that are not necessarily regulating hormones directly, but they do produce and respond to hormonal signals. So hormonal release is in response to a stimulus. Typically, it's a reflexive stimulus. You can have release through 3 different mechanisms. You can have hormonal release in response to a hormone. So a hormone binds its receptor and causes another hormone to be released. That's hormonal stimulation. Humoral stimulation is a hormone being released in response to a change in something in the blood. So a change in blood glucose concentration or sodium concentration in the blood can cause another hormone to be released. Or nervous system stimulation. So you can directly stimulate a hormonal tissue and cause that hormonal tissue to release a hormone. So that endocrine tissue was responsive to the nervous tissue. The distinctions here clear? Okay, so know these 3 things. Okay, so like hormonal stimulation. The anterior pituitary sends out hormones that then act on target tissues and make those target tissues send out hormones. So thyroid stimulating hormone is released from the anterior pituitary into the bloodstream. It then goes to the thyroid gland, where thyroid hormones, T4 and T3, primarily T4, are released into the bloodstream and then they act on some tissue. The pancreas responds by releasing either insulin or glucagon in response to blood glucose levels. Blood glucose levels get too high, what's released? Insulin. Blood glucose levels get too low, glucagon's released. Sympathetic nervous system innervation of the adrenal gland was one of the 4 sympathetic pathways we talked about last Monday, right? On Monday? That's the last time I saw you guys? Yes. I don't have my glasses. I can't see anybody today. This is an example of nervous system stimulation of the endocrine system. So the adrenal gland is an endocrine gland. It's stimulated through the sympathetic nervous system through this adrenal medullary pathway to release epinephrine and norepinephrine, which are dumped into the bloodstream. So here they're acting as hormones, not necessarily as neurotransmitters. So it depends on where and how that epinephrine and norepinephrine is released. If it's being released and acting on a postsynaptic cell, then we call it a neurotransmitter. If it's being released and dumped into the bloodstream so that it can act on a cell that's far away from the site of release at the adrenal gland, then we call it a hormone. Same molecule, different name depending on its action. So hormones travel through the blood system. They travel through our cardiovascular system, so they're circulating. Steroids are one type of hormone that circulates in our body, but a steroid is a lipid, right? And our blood is made of primarily what? Water. No. What is happening?
Hydrophobic.
It's hydrophobic, exactly. I don't know why this is being weird. Try again. What?
Oh, we were just talking about why our computers die so quick. Because there's so many tabs open on them.
Okay, you leave me alone about my tabs. I can't help it. I know it's a problem.
I'm not judging. I have the same issue. My husband judges.
That's mild for me, okay? All right, back to the slide we were on here with circulating hormones. Okay, so we're talking about steroids. Steroids are lipids, but our body is full of mostly water in our blood system, right? So these steroids, which are lipids, don't dissolve in water, right? They don't dissolve in our blood. So how do we move them through our blood system? That's a really good question. It happens every day, right? How do we move lipids, a hydrophobic molecule, through our blood system, which is with what? I'm sorry, water? No, I can't.
No,
I
it's
can't.
not lipid bilayer.
You're saying that how do we move it?
Yeah.
Since
It's hydrophobic,
right.
and our blood is made primarily of water, which is a polar molecule. So how do we move a non-polar molecule through a polar solution? It attaches to something. Yes, ma'am. All right, Kat's on to something here. It attaches to something. It attaches to a carrier protein. So typically, steroid hormones are carried via a carrier protein through the blood system.
So is it like enclosed in this protein, or is it attached?
It just attaches.
It's kind of like how do I say it?
It's a chaperone.
Like a virus?
It's like, no, it's not like a virus. No. It's a chaperone. No, it's like, yes, it's like carrying your kids through the museum. Your kids are not going to be allowed in the museum alone because they will wreak havoc on the place. Instead, you go as a chaperone. So you walk in holding their hands and say, they're with me. I'm taking responsibility for these hooligans.
Okay.
And then, not that your kids are hooligans, mine is, but okay, so our both of us, all right, so we're standing there taking responsibility for our hooligans, and we're allowed to go into the museum with our kids. We're carrying them through the museum so they get where they're going in an appropriate time frame without tearing everything up. So these are, yeah, it's not a virus. It's not a virus. Okay, so these are lipid-soluble molecules. What does that mean about how they enter cells? Yeah, they're small lipid-soluble molecules, so they can wiggle through the phospholipid bilayer. So they can transfuse or diffuse across the membrane. Okay, so this is primarily your sex hormones and stress hormones that are steroid-based hormones. So what does this molecule look like? These 4 rings? What does this look like? What is it? Yeah, but what is it? What does this look like? It's an all. It's an all, but which one? Cool. Say it. Cholesterol, yeah. So these steroids are derived from cholesterol. Do you remember how last semester we were talking about how people say, oh, cholesterol's so bad for you? Don't eat any cholesterol. Well, you don't get your hormones. We need cholesterol. That's how our steroid hormones are generated. So we need these steroids because they are things like estrogen and progesterone and testosterone and cortisol. We need them, right? So you need cholesterol to make them. Calcitriol is sometimes included, but it's not really a steroid-based hormone. It's a sterol. There's a difference. So we're not going to call calcitriol a steroid hormone. You guys remember what calcitriol does?
That last few.
It tries to tone down, or tries to break down bone to release calcium. Doesn't break down calcium. Tomato. Not exactly. Okay, so another characteristic of circulating hormones is that they're small molecules. These biogenic amines that we talked about in the sympathetic nervous system, epinephrine and norepinephrine, they can also be considered hormones if they're dumped into the bloodstream and they're acting at a tissue distal from their release site. So thyroid hormone is another one. So melatonin, thyroid hormone, epinephrine, norepinephrine. These are all biogenic amines or monoamine hormones. What's happening? What have I done now? Am I taking screenshots? I think I am. Sorry, y'all. But these can dissolve in water. They have polar ends to them. So they can dissolve in water. That means they can be carried through the bloodstream without a chaperone protein. Most of our hormones are characterized as proteins, not just small molecules. These proteins generally have areas of relative electronegativity so that they can dissolve in water, at least partially. So they're water soluble. They're usually small to medium-ish chains of peptides, and they can fly right through our circulatory system no problem because they are polar and our solvent is polar. So insulin, glucagon, antidiuretic hormone, growth hormones, all considered protein-based hormones. So those are your 3 categories: steroids, monoamines, and proteins. We can have signaling molecules that don't circulate in the blood but are still considered hormones. This is called autocrine signaling. When one cell releases a hormone and then that hormonal signal acts directly on the cell that it was just released from. So there's always an exception to every rule, right? Because I just told y'all that a hormone is defined as a molecule that acts distal from its release site, except when it doesn't. Autocrine signaling is when the cell that has released the molecule is acted upon by that exact molecule. Typically, this is a negative feedback, so we can stop that cell from releasing more hormone. It doesn't have to be every time. If that hormone is acting at another tissue, we call it paracrine signaling. So autocrine and paracrine are 2 terms you need to know. Okay, so last semester when we went through lipids, we talked about a bunch of different categories, right? We talked about triglycerides and phospholipids and steroids, and there was a 4th on your slide that I told you don't worry about. Eicosanoids and prostaglandins are that 4th category that we weren't worrying about then, but we're going to worry about now. Eicosanoids are locally or autocrine acting hormones made from fatty acids, made from phospholipids. They are synthesized via enzymatic actions, and they are eventually made into prostaglandins. Prostaglandins are molecules that evoke an immune response, an inflammatory response. So anytime you're injured, you have prostaglandins released, synthesized at your site of injury. Prostaglandins synthesis is where aspirin and NSAIDs act. So we start out with a phospholipid, right? Phospholipid. Because that's a what? What is this thing called? Phospholipid bilayer. Okay, so an enzyme called phospholipase A2 breaks down those phospholipids from the plasma membrane into something called arachidonic acid. So it's just a fatty acid. It's a specific fatty acid. That fatty acid is then metabolized into prostaglandins or thromboxanes via COX, cyclooxygenase, 1 or 2. Those are the enzymes that make prostaglandins and thromboxanes, and these are the molecules that are going to evoke that inflammatory response. Have you ever heard of a COX inhibitor? Have you ever taken Tylenol?
Yes.
COX inhibitor.
I take some today.
Me too. After that headache of mascara.
Yeah, same.
Yeah, you too? Mascara all over the place? Every morning. COX inhibitors act here to prevent the conversion of arachidonic acid to prostaglandins. That's how pain relievers work. Now, COX enzymes, you have to keep making these, because, especially if you chronically take NSAIDs, because once an NSAID has bound to a COX enzyme, it is permanently deactivated. You can never reactivate that enzyme. Typically, a drug will bind to something and then eventually it'll unbind and it'll be metabolized or broken down and excreted, right? Not so with COX. COX inhibitors permanently deactivate that enzyme. So that enzyme, the whole thing, is now useless and it has to be broken down. Great for pain relief, but it does put some pressure on the genes that code for COX because you have to keep synthesizing it over and over and over again. Okay. The arachidonic acid can also be metabolized into leukotrienes. And this is through glutathione S transferase. Let's actually write that correctly. And in the liver. So those are the three primary products you get out through conversion of arachidonic acid to eicosanoids. Okay. So lipid-soluble hormones, we already talked about this, must have a carrier protein to get through the blood because they don't dissolve in blood because they are nonpolar. And blood is a polar solution, right? It needs a hydrophilic molecule to dissolve in it. Hydrophobic molecules will not. The carriers are water-soluble proteins, typically made in the liver, and they protect the hormone. They carry it through the museum, get it to where it needs to go. But the binding between these is temporary. So once you get those kids through the museum, you can just let them out the front door and let them run. The carrier protein just lets the hormone loose where it needs to go, and it doesn't ever have to go with it again. Most of your hormones floating around in your blood system are bound because it's really energetically unfavorable for a steroid hormone to be unbound to its carrier protein in your blood. It only unbinds when it gets close to its target tissue so that it can wiggle through the plasma membrane, because remember it's nonpolar, so it can get through that phospholipid bilayer and get into its target tissue to bind to its receptor.
How will it know?
It doesn't really know. This is kind of a random thing. It just kind of binds and unbinds. It doesn't know. It's not sentient. So it seems like I'm making it sound really, really controlled, right? It ain't. It's kind of a mess. Hormones are kind of a mess. Have you ever seen a 13-year-old girl? It's a mess. Totally dysregulated. Yeah, I've been a 13-year-old girl. It's totally dysregulated. But the water-soluble hormones, they can float through the blood system just fine. Once they get near their target tissues, they'll diffuse into the interstitial fluid and they'll bind to their receptors just fine. Occasionally they'll use a carrier protein because sometimes they can be broken down really easily, so the more susceptible ones may use a carrier protein, but they don't have to because they are polar molecules. They can dissolve in a polar solvent, blood. Okay, so the blood concentration of a hormone depends on how fast you can make the hormone and how fast you're breaking down the hormone. So hormone release is positively correlated with blood hormone levels. The more you release, the higher your blood hormone levels. Hormone elimination, however, can occur through different mechanisms. So release typically happens on one side. Elimination depends on the hormone. It can either be degraded in the liver, it can be eliminated in the kidneys, or it can be metabolized down by other proteins. That's not as common. The faster you eliminate the hormone, the faster you have to make the hormone if you're trying to keep consistent hormonal concentrations. Does this make sense? We can measure levels of the hormone by the half-life. What is half-life? Does anybody know?
How long it takes for half of it to disappear?
Exactly. So half-life is how long it takes for approximately 50% of a molecule to be broken down. So if I want to break down all of a molecule and the half-life is 24 hours, how many days will it take to break down virtually all of a molecule? 2 days will give me 50% gone on the first day, and then there's 50% remaining, so then we'll have 25% gone on top of that. So we'll still be remaining 25% of the original amount. So then by day 3, we'll be at 12%. By day 4, we'll be at 6%. By day 5, we'll be at 3%. So yeah, between 5 and 6 days to get virtually all of the hormone out of your bloodstream if the half-life is 24 hours. Because every 24 hours, you lose half of what you've got. The half-life itself depends on how efficient the elimination process is. Water-soluble hormones have a shorter half-life, and steroid hormones typically have a longer half-life.
Is this why when people like bodybuilders, when they take those steroid stacks and stuff like that, you have to give your liver whatever 3 to 4 months to clear out, or other than to just shut off, or not shut off, but they'll just mess up everything.
Mm-hmm.
Okay.
Because you have to metabolize them out. And some of them can take weeks to metabolize.
Yeah.
If you have a half-life of weeks and weeks, yeah, you gotta give it some time, because okay, so if your half-life is 2 weeks, then that means 4 weeks to get to 25%, and if, see, 6 weeks to get to 12%, 8 weeks to get to 6%, and 10 weeks or 2.5 months to get to 3%, and it's still probably detectable at 3%. So, yeah, that's why you need months. Or are you peeing on a steroid test? Okay. Typically, those steroid hormones have a carrier protein protecting them, prolonging their half-life. So that's partially why. Also, they can be stored in adipose tissue.
I was about to say, they make you big if you're on it for prolonged. Like someone that has to take steroids when they make you big.
What do you mean by big?
Like when they make you gain weight because they store in other parts of your body.
Not necessarily. Those molecules are so small. But they can influence metabolic processes that can cause you to either retain fat or to build more muscle. So, one is a good big and the other is not. Like big versus swole.
Yeah.
You want swole, you don't want big. Okay. So those lipid-soluble hormones are going to diffuse across target cells. They are lipophilic, so they wiggle straight through the phospholipid bilayer. Their receptors are typically in the cytosol or in the nucleus. If the receptor's in the cytosol, often that receptor, once it's bound by the hormone, will then translocate to the nucleus, and it'll still act in the nucleus. It'll still be the end target most of the time with these cells. When the hormone binds to its receptor, we call it an HRC, or a hormone receptor complex. That hormone receptor complex can then become a transcription factor that we call a hormone response element. A transcription factor either upregulates or downregulates the transcription of certain genes. So it goes into the nucleus, down to the DNA, and it acts, resulting in either increased or decreased transcription of a certain gene. What was transcription?
Can they copy
Copy the what?
the DNA
Into.
RNA?
Yes, copying the DNA into RNA. That's transcription. So that's the start of making a protein, right? We've got to first get that DNA copied over into the RNA code for us to be able to make a protein out of it. So these hormones modulate that activity, either increasing or decreasing it. This hormonal response element acts as a transcription factor. Either increasing or decreasing transcription of a gene. That increasing or decreasing transcription of a gene has effects on increasing or decreasing translation into proteins. So how many functional proteins do you have out of that will change based on the hormonal stimulation. So here's a pretty picture. Got a hormone coming. So here it is, bound to its carrier protein. It unbinds, goes into its target cell, forms a hormone receptor complex, then goes into the nucleus as a hormone receptor complex that can act as a transcription factor. Once it binds to the DNA, we call it a hormone response element, which is just a transcription factor. That then changes the transcription of that DNA or RNA. So just to be clear, a transcription factor does not have to be a hormone response element. It does not have to be that. It can be another separate protein. But a hormone response element can be classified as a transcription factor. Does that make sense? Not all transcription factors are hormone response elements. But all hormone response elements are acting as transcription factors. Just want to be clear. And what happens here depends on what the hormone is and what the target tissue is. Okay. So that was the steroid hormone. Let's talk about the water-soluble hormones. So we had those biogenic amines, and we had protein molecules that were our water-soluble hormones. So the water-soluble hormones are going to use the membrane receptors. These typically are not going to diffuse across a membrane because they're a little too big to do that. Remember, there were some very, very small polar compounds that could wiggle through the membrane, but For the most part, these are going to be way too big to be able to wiggle through a membrane. So they're going to have to bind to a receptor on the outside of the cell. So the surface of the cell is expressing a receptor for this hormone. Once that hormone binds to its receptor, some change is going to happen inside the cell on the other side of that phospholipid bilayer, and it's going to initiate some signal transduction pathway. The hormone is what we consider the first messenger. So what does that tell you we're going to have? If we have a first messenger, there must be a second messenger. Yes. So the second messenger is going to be that G protein. So the first messenger, the hormone, binds to its receptor, and it activates a G protein inside. So when that G protein is activated, a GTP is bound to it. So it kicks off a GDP. That GTP is bound to it. And then it can go and do something else in the cell. Typically, it goes down to another enzyme. So like adenylyl cyclase or phospholipase C or PKM zeta. There's a whole long list of them that this specific, that a specific G protein can bind to. And it depends on the G protein and the cell it's in and the receptor that was stimulated as to what downstream enzyme will be activated. So once that enzyme is activated, then the second messenger system will kick off, and it will modulate the cell's activity by starting a kinase cascade or opening an ion channel somewhere else. And again, it depends on which tissue you're talking about, which hormone was bound into which receptor it bound.
What's GCP?
It's guanosine triphosphate,
And then the GDP?
diphosphate. So in order to deactivate that G protein, it has to be dephosphorylated from a triphosphate to a diphosphate. Yep. And then that GDP stays bound until that receptor is stimulated again. And at that time, the GDP is kicked off and a GTP will bind, and that GTP is essentially, it's the gas in the tank to carry that G protein to the first enzyme to activate the kinase cascade. So here we are. Here's our receptor. Our hormone has bound to it. We kick off a GDP. Here now I'm answering the question with a picture. A GTP binds to it, and that G protein is activated, and it can come down and it can activate something else downstream to initiate a signaling cascade. So this is the kinase cascade. Some of these kinase cascades are really, really small, just like 3 or 4 enzymes, and some of them are like 30, 40, 50, 60 enzymes. It can get really complicated really fast because there's a lot of crosstalk between these. So you can see how a hormone stimulating one receptor, if it eventually activates 30 or 40 different kinases, enzymes, then it can have widespread effects across multiple cells of a tissue, causing widespread effects across the body. So hormones are easily amplifiable signals. This is another difference between the nervous system. The nervous system signals are not so easily amplifiable. Okay, so when that GTP binds to a G protein and activates it, if it comes down and activates adenylyl cyclase, then cyclic AMP will be the enzyme that's activated next. Cyclic AMP can activate a protein kinase. That protein kinase then phosphorylates another molecule, which could be another protein kinase, which could then phosphorylate another protein kinase and another protein kinase, and eventually it'll get to a target molecule that'll be phosphorylated, and it'll either activate or deactivate that molecule. In general, that will be a molecule that will change gene transcription, either turning it on or turning it off again. You seeing a pattern here? We're always trying to get down to the genes. We're always trying to change what's happening with gene transcription because proteins are the way we do things in the body. There's no point in releasing a hormone if it can't get down to the level of the genes and change fundamentally what's going on with our protein expression. So our proteins are doing the job. The hormone is saying we need to change the job that's being done. So in general, we're going to get down to that level and we're going to change gene transcription. So here is an example of adenylyl cyclase being the kinase that's activated. So GTP is bound to our G protein, which means it's activated. It's going to bind here at adenylyl cyclase. We're going to use an ATP. We're going to activate cyclic AMP. cyclic AMP can then come and it can bind to another protein and activate that protein. So this protein kinase, here it's being called protein kinase A. It can be lots of different protein kinases. There are dozens and dozens of them. Protein kinase A is a specific name of a protein kinase as well. So I don't like them calling it protein kinase A, but it's okay. However, you can have multiple downstream effects until you get into the nucleus and start changing gene transcription. You don't have to do that every time, but in general, that's where we're headed. Okay, phospholipase C is a specific enzyme that can be activated after norepinephrine is released, or epinephrine. So epinephrine is released, it binds to its receptor, and a G protein becomes activated. The GDP is kicked off, a GTP is bound to it. That activated G protein can then activate phospholipase C. Phospholipase C will then split a molecule called PIP2 into DAG, diacylglycerol, and inositol triphosphate, or IP3. These are two signaling molecules, second messenger signaling molecules that can initiate huge changes inside of a cell. I mean, change huge kinase cascade signaling mechanisms by just activating these. So protein kinase C can be activated by diacylglycerol, DAG. It can then go on to phosphorylate other molecules downstream that can eventually change gene transcription. IP3 is a second messenger, and this is an example of where we don't change gene transcription directly. IP3 is a second messenger that causes an increase in calcium levels in the cytosol. What does that mean for us if we're increasing calcium levels in the cytosol? Or in the cytosol, we're inside the cell, so we're not in the blood. It can start to depolarize the cell. It changes the membrane potential of the cell if it releases enough. Where is calcium stored inside the cell? Where at? Inside the cell. Inside a single cell of any tissue, there's a storage reservoir for calcium. Which one is it? It is the smooth endoplasmic reticulum. Oh, because it says it right there. The smooth endoplasmic reticulum. Smooth endoplasmic reticulum can be stimulated by IP3 to release calcium. Calcium can then act as a third messenger. So it can go down and activate other kinases, and then it can go down and activate other ion channels, further depolarizing the cell or repolarizing the cell, depending on the channel that's activated. Starting to get complicated here? This is nothing. Nothing. So here's that activated G protein. It's come down here and it's activated phospholipase C. Phospholipase C then activates PIP2, which breaks apart into IP3 and DAG. Here's where they split up, and they do two different jobs at once. So DAG comes over and activates protein kinase C, which changes some downstream signaling thing. Boom. IP3 comes down to the smooth endoplasmic reticulum, activates the release of calcium. Calcium comes in activating calmodulin and other protein kinases, which can be activated by both calcium and calmodulin. And it can also activate ion channels. So you can see how this gets real complicated real fast, and you start getting a whole lot of different effects. So you've got ion channels opening, so we're changing the membrane potential. We've got these other kinases activated, so they're coming down, probably activating more kinases. And eventually, something is coming down here and changing gene transcription. Probably of multiple genes, because these are different enzymes that are being activated. Different enzymes probably acting as different transcription factors for different genes. It's widespread effects from just one hormone binding one receptor. Wild, huh? No wonder 13 was hard. All right. So this is how we can get so many different endpoint results from one molecule binding. Different signal transduction pathways being activated from one molecule binding its receptor. The enzymes can be activated or inhibited. They don't all have to be activated. Here I've been showing you activation, activation, activation. They don't have to be. They can be deactivated. They can be phosphorylated or dephosphorylated. And phosphorylation doesn't always equal activation. Sometimes you phosphorylate something and that shuts it down. We can stimulate growth or cell division. We can stimulate the secretion or release of specific molecules from a cell. We can change the membrane permeability. We can change the membrane potential. And we can contract or relax muscles. Sometimes all with the same hormone. Okay, not all of these with the same hormone, with a couple of them. Okay, so those intracellular enzyme cascades are a mechanism of amplifying the signal. So I showed you several different pathways that can be taken by signaling molecules that were all derived from just one hormone binding its receptor. But then every time those signaling molecules continue down their path, their signal is amplified. So just because there are two different signaling pathways that develop from the metabolism of PIP3 doesn't mean it just stays two linear signals. It amplifies out like a tree. Luckily, we have redundant mechanisms to control these pathways. So we can phosphorylate just as fast as we dephosphorylate. We can inactivate just as fast as we activate. So your cell can come back and shut down these signaling cascades if necessary. So again, this is always a balance. Just because you have hormones binding doesn't necessarily mean that those hormones are going to exert the activity that they're intended to. You have to have a sufficient enough signal in order to get that end result of changing gene transcription. If there is an opposing signal that is stronger, your hormone can bind all day long, but if that receptor is being cut off at adenyl cyclase or at phospholipase C, where those downstream effectors are being shut down, then you'll never get to the end point that your hormone's trying to achieve. Does this make sense? I know that was a lot to throw at y'all. Then it's a lot of non-specifics. I don't want it to be specific because it really is very case-by-case dependent. Which hormone, which receptor, which cell, which subset of cells within a tissue. What signaling molecules are already present there? So just understanding the general themes of that. These signals can be amplified, that there are multiple pathways that are affected within a cell after binding a single hormone to its receptor. That's sufficient for this. Okay, let's see. The degree to which a cell responds to a hormone depends on the number of receptors that are expressed for that hormone and the responsiveness of that cell to other hormones. So there are always antagonistic effects to everything that's happening in our body. Why do we have antagonistic effects to every agonist in our body? To balance, which is called homeostasis. Our bodies are always trying to stay in homeostasis. So we always have opposing effects. You're trying to go up, okay, we're going to push you back down again. Trying to go left, all right, we're going to pull you right. Always trying to keep you in homeostasis. So if you want to initiate a response and you're a hormone, you've got to act on a cell that has a sufficient number of receptors so that you can dump enough hormone onto enough receptors that they all get activated at the same time so that the signal inside that cell is large enough to overcome the opposing effects. So how do we get receptors on a cell membrane? What is a receptor? A protein? Yeah. How do we get a protein onto a cell membrane? Binding? What's that? Integrate? What do you mean?
Doesn't it?
They're made in the ER, which one?
Rough.
Rough. Yeah, so we have to make that protein, right? We have to make that protein. So we have to transcribe and translate. So we transcribe the gene that codes for that protein. Then we translate it into a protein in the rough endoplasmic reticulum. Then we process it. And then we translocate it into the plasma membrane. So remember, it can be packaged into a vesicle and then exocytosed, or it can be packaged onto a membrane of a vesicle, and that whole membrane can be inserted into the cell membrane. So once we get it there, then it can be acted upon by a hormone. We can upregulate or downregulate the number of receptors that are there. So if we upregulate, then we increase the sensitivity to that hormone because we have more receptors that can bind the hormone. This happens often when blood levels of a specific hormone are low because your body's like, oh, wow, we're not getting a whole lot of that signal. Let's provide more opportunities for that hormone to bind. Downregulation is decreasing the number of receptors, and this typically happens if you have too much of a hormone. Your body's saying, hey, we're a little overwhelmed here with all that hormone. Let's stop responding to it. Let's take away some of those responders. It's like when I'm inundated with like 100 emails in 24 hours and I just close my inbox. I'm like, I can't respond right now. Shut it down. Roll it back. Either of these can occur through developmental changes. They can occur with different patterns of cellular activity. They can occur at different stages in the cell cycle. But they always occur in response to the level of hormone that is present. Too much hormone, you downregulate receptors. Too little hormone and you upregulate receptors. Downregulation decreases sensitivity, upregulation increases sensitivity. All right, this is an example. Upregulated hormone receptor. So those little Y's here, that's a receptor. So here's your hormone. You've got a little bit of hormone binding these receptors. You've got a whole lot of receptors, a whole lot of opportunity to bind. Downregulation occurs when you have a whole lot of hormones and you, as a cell, are getting overwhelmed with hormonal signaling. So you only have a few receptors here so that your degree of response is lower. Okay. Hormones can have multiple different effects at the target cell. So you can have hormones that are working in synergy with each other, so they're adding together to increase the effectiveness of whatever process they're trying to initiate. So one hormone will reinforce another. So like estrogen and progesterone typically reinforce each other. You can have permissive hormonal interactions where one hormone has to be present in order for another hormone to work. So like oxytocin does not, so we talked about that as a positive feedback mechanism, right? So oxytocin does not work in the absence of prolactin. So you have to have prolactin and oxytocin to generate the milk letdown reflex. Or they can antagonize each other. Like insulin brings down blood glucose levels and glucagon raises them. It mobilizes glycogen stores from the liver. So this is one hormone acting in opposition to the other. Okay, we're going to stop there for now. Yeah. And we'll pick up pituitary gland once you guys take a break. Cool? Sound good? You guys need a break?
Yes.
We're going again? Good. Okay, so let's move up to the primary hormone regulator. This is the pituitary gland. So the pituitary gland is regulated by the hypothalamus. We remember where the hypothalamus is, right? Can you give me the general organ? In the brain. Anybody know more specifically?
The thalamus.
But where specifically, if I'm looking for a landmark, what do I find?
Superior to the hypothalamus.
There's a structure right underneath it.
The gland.
Looks like this. That's the one. Kia's got it. Under the above the optic chiasm, right above the optic chiasm that feeds directly into the hypothalamus. You guys remember what the optic chiasm is, right? What is it? That's where the optic nerves decussate.
Yeah.
All right, so that's where we're at as far as the hypothalamus. The hypothalamus is sitting essentially in the sphenoid bone in the sella turcica, that little dip, the little cavity on the base of your skull. It is a pea-sized gland. It's not a gland, it's a brain region that connects to the pituitary gland through the infundibulum, which is a tract. What is a tract? Bundle of axons, yeah. And we can divide it into the anterior and posterior pituitary glands. So the posterior pituitary is the smaller part of the pituitary gland, and the hypothalamus projects through the infundibulum into the posterior pituitary, sending signals from two specific nuclei from the hypothalamus. The PVN, or paraventricular nucleus, PV, let's actually write the right letters, PVN, and the supraoptic nucleus. The tract that projects via the infundibulum is the hypothalamo-hypophyseal tract. Well, yeah, I know, right? So posterior pituitary, hypophyseal. The synaptic knobs are in an area of the posterior pituitary called the pars nervosa. What's a synaptic knob? The end of the axon. That's where that's the action site, right? That's where all the cool stuff's going to go down. Neurotransmitters going to be released. Okay, so this is a subset of cells inside the posterior pituitary. So here we are. Here's your posterior pituitary. Again, not testicles. This is in your brain. So here's the paraventricular nucleus projecting to the posterior pituitary, and the supraoptic projecting to the posterior pituitary. Paraventricular makes oxytocin. Supraoptic makes antidiuretic hormone. Huh? What? Antidiuretic hormone? It's on your slide. It is right there. On your slide. Yeah. So the posterior pituitary is going to store these hormones. So this is where the cell bodies are up here, the somas, the cell bodies. So we make the hormone there in the hypothalamus, and then we transport it down these axons. What type of transport is that called? We're going down away from the cell body. Transport mechanisms. Transport, not that kind of transport, but down an axon. There were two ways we can transport things up and down an axon. Is this up or down? Down. Interrograde transport. These are stored in the posterior pituitary, and then they'll be released from there when the right signals are received. Okay, so it's the posterior pituitary. The anterior pituitary contains the hypothalamo-hypophyseal portal system. This is the blood vessel network that allows us to dump these hormones into the bloodstream. You have a primary, secondary plexuses. The primary plexus is associated with the hypothalamus, and the secondary is associated with the pituitary, specifically on the anterior pituitary. And the portal veins will drain the primary plexus and transport to the secondary plexus. So this is how we get those hormones from the pituitary into the rest of the body, via this portal system. Here it is. So primary up here, secondary down here, and here are the portal veins in the middle. So this is a list of hormones made by the hypothalamus and by the anterior pituitary. Know these hormones. We're going to talk about most of these as we go through this lecture. Just giving you a heads up, this is a good summary of those hormones. Know what these are.
So do these go from those glands to the other glands that need to, or do those other glands produce their own?
Nope, they're released from here, go into the portal system, and they travel to the tissues where they're going to bind to their receptors. I know it's hot in here. I'm so sorry. Don't be anxious. It's okay. Aw, one note. Why is it so hot? I mean, we could. Would y'all rather move to the lab? Are y'all dying? We can vote. Who wants to move? It's just you, Eddie.
No, I was just saying this is options. I mean, last time, didn't
Hold on, one thing is the link.
Okay. All right, so the posterior pituitary, we just talked about that up here. Okay, so here's your portal system. All right, so it's back to the posterior pituitary. So this is where oxytocin and antidiuretic hormone are made. So the hormones are made in the hypothalamus by neurosecretory cells. They're then transported anterogradely down the axon to the posterior pituitary. They're transported through fast transport. Fast transport means they're transported in vesicles. Put something in a vesicle, you can move it quickly. You package something up, send it to UPS, it goes fast, right? Faster than FedEx. So these are fast axonal transport, anterograde transport via vesicles. They hang out in those vesicles in the synaptic knobs. And then they're released from the pituitary when they get the appropriate signals. So vasopressin or antidiuretic hormone is made in the supraoptic nucleus, and it decreases urine production, stimulates thirst, and constricts blood vessels. Why would you need to do this?
If you're dehydrated.
We need to retain water. Antidiuretic hormone. Oxytocin, made by the PVN, primarily functions in uterine contraction, milk ejection, and emotional bonding. This happens between mothers and their babies and between sexual partners. Oxytocin plays a role in emotional bonding. That's why you may or may not like your partner. Thanks, oxytocin. It's true. You're looking at me like it's not true.
There's a study linked to oxytocin and postpartum depression now too.
Yeah, there are lots of studies linked to oxytocin and postpartum depression.
Because the going through labor drains all your oxytocin, so it bonds out and it causes the postpartum depression.
Exactly, you can't bond with your baby. And you can't bond with anybody, really. Okay. The hypothalamus hormonally stimulates the anterior pituitary, telling it to release its hormones. It secretes regulatory hormones that then travel through the portal veins to the anterior pituitary, which is that secondary vascular plexus, and tells the anterior pituitary, hey, here's your signal, release your hormones. So it's a 2-hormone step. First set of hormones is made and released from the hypothalamus, dumped into the portal system, goes down to the anterior pituitary and tells the anterior pituitary to release its hormones, which will then go to its target tissues in the body. Cool? Cool. Okay, so hypothalamic hormones. So these are primarily releasing hormones. So thyrotropin releasing hormone, prolactin releasing hormone, gonadotropin releasing hormone, corticotropin releasing hormone, and growth hormone releasing hormone. So these are all hypothalamus. Hypothalamus releases releasing hormones. They also release inhibiting hormones like prolactin inhibiting hormone or growth inhibiting hormone. These stop the anterior pituitary from secreting prolactin and growth hormone. The anterior pituitary, once it receives the right signal, will then start releasing the appropriate hormones. So if thyrotropin releasing hormone is sent from the hypothalamus to the anterior pituitary, then it will release thyroid stimulating hormone, TSH. So TRH makes TSH be released from the anterior pituitary. This causes a release of thyroid hormone in the form of T4 primarily from the thyroid gland. Prolactin releasing hormone, or prolactin is stimulated when prolactin releasing hormone reaches the anterior pituitary. It's inhibited by prolactin inhibiting hormone, and it causes milk production, uterine contraction, and mammary growth, and bonding. Again, it's not really love. It's just hormones. Avatar with the tail. Oh gosh, that's so weird. You don't have to go there. Adrenocorticotropic hormone, or ACTH. This is also called corticotropin, and it is released when CRH, corticotropin releasing hormone, is released from the hypothalamus. This causes your adrenal gland to release corticosteroids. So you guys seeing a pattern here? Yeah, okay. Again, scientists are not super creative. All right, so the anterior pituitary hormones continued. There are two gonadotropins in both males and females. This is follicle stimulating hormone, or FSH, and luteinizing hormone, LH. Gonadotropin releasing hormone is released from the hypothalamus. It tells female organs to regulate ovaries, ovarian development, and secretion of estrogen and progesterone. In males, it regulates the testes to produce sperm and testosterone. Same hormones, different actions depending on the anatomy. Form follows function. Growth hormone, or somatotropin, causes the liver to secrete something called IGF, insulin-like growth factors. IGF-1 and IGF-2 are two similar but distinct proteins that are released, hormones that are released from the liver. They act with growth hormone to stimulate cell growth and division, primarily of skeletal and musculoskeletal tissues. So here's a good summary of those hormones we just talked about. So if you know this chart, you're solid. It's cool, no big deal. It's really not that complicated. It's really, truly not. This is just graphically representing that for those visual learners. It's the same thing I just said to you. Literally the same thing I just said. It's okay. Deep breath. Don't die. Okay, so we take out the pituitary gland. So pituitary gland is a common place for brain tumors to occur. So we take out the pituitary gland, you go in through the nose, you yank that sucker out, because remember it's in the base of the skull at the sella turcica. It's just a little you can use like a melon baller. It's not really a melon baller, I'm being a little sarcastic. But up through the nose, pop it out, but then you don't have all those hormones. So you have to replace all of those hormones using exogenous hormone replacement therapies, or else your whole body can't function.
Would they replace the pituitary hormones, or the ones that it would stimulate?
The pituitary hormones, often.
The rest of your body's still.
Yeah, well, it depends. So like thyroid hormone, you can replace that directly with T3 and T4. You can also replace TSH, so Synthroid replaces TSH. So you have options. You don't have to do one or the other. Most of the time we have options for both. Okay, so growth hormone, released in response to growth hormone releasing hormone. There's also growth hormone inhibiting hormone.
Isn't this what pregnant ladies have to inject with? Like the ones that are doing IVF and surrogating?
No. You wouldn't do growth hormone. No.
I know there's a reason for growth.
Yeah, there is. And usually it's a genetic reason. Okay, so the amount of growth hormone released by the hypothalamus is determined by how old you are. Obviously, children are in a growth phase, a rapid growth phase, so they're going to have more growth hormone releasing hormones secreted. The time of day is important. Typically, a lot of your growth hormone is at night. Rest and digest also means growth. You don't typically put a lot of energy towards growth during the day when you're performing activities, because your body needs that energy for other things. Nutrient levels, so if you're not eating sufficiently, you're going to have decreased growth hormone. This is why you can't build significant amounts of muscle mass when you're in a caloric deficit. And stress and exercise as well. So if you're exercising a lot or you're under a lot of stress, nutrients are being diverted elsewhere. So you're not going to get a whole lot of growth hormone releasing hormone because your body can't do anything with that. It doesn't have the building blocks to utilize the growth hormone properly. Okay, so again, growth hormone stimulates insulin-like growth factor being released from the liver. Insulin-like growth factor has a longer half-life than growth hormone, so it can act for a longer period of time and at a wider variety of tissues. All cells have the receptors for these two hormones, but they have different densities. The hormone will change protein synthesis, and it will increase cell division and cell differentiation of stem cells. So it makes your stem cells mature into their terminal cell types. It stimulates nutrient acquisition. So where would we get nutrients from? Food, but in our bodies. Where are these hormones going to act? They can't go act on a cow. In your body, where can you get nutrients from? Liver stores.
Fat cells.
Glycogenolysis. We're going to break down glycogen from the liver. Because glycogen is what? What is it? Say it. Say it louder. I heard it. It's sugar, but what specifically? What is it? It's a polysaccharide, but of what? What is the monomer? Glucose. Yeah. So it's going to mobilize glycogen from the liver so we can break it down into glucose molecules so we can use those glucose molecules for energy. Also stimulating the liver to undergo gluconeogenesis, which is making glucose from other macromolecules. Glycogenesis is inhibited because we don't want to take those glucose molecules and put them into a polymer and store them. We want to keep them in the bloodstream for use. So we don't want to make glycogen. We want to convert the glycogen to glucose. Glucose is usable, glycogen is storage. These two hormones are all about getting usable energy going. Lipolysis, breaking down your adipose tissue, breaking down those triglycerides into free fatty acids so that you can undergo beta oxidation and utilize that energy. IGF and growth hormone will do that. But lipogenesis, making triglycerides and packaging them into adipose tissue, is inhibited because we don't want to store the energy. Again, we want to take it out and use it. If you're in a growth phase, you need energy. So all of this is about trying to get energy out. So here we're releasing growth hormone, releasing IGFs. They're going into the blood. We're trying to get more glucose from glycogen stored in the liver. We're getting fatty acids dumped from adipose tissue. We are moving amino acids into our tissues, primarily our musculoskeletal tissues, because we're in a growth phase. We're trying to get those tissues to grow. High levels of growth hormone up until about 20 years old. And then it just kind of plummets because you're not in a growth phase anymore, right? We are done growing, at least up. We might grow wider, but not up. So we don't need growth hormone anymore. It's higher near the ends of the day. At night, when you're sleeping, when you're resting, that's when you're going to have the most growth. We don't have a whole lot. We're kind of past that phase. How many of you are under 21? Oh, just get out of here. Just get out of here. All right, y'all are still squirting out some growth hormone. The rest of us, it's over. It's over. Yeah. Okay, so if you have a... No.
For sure done by the time you're 30. Like that's when you start aging at that point.
Can you not? 26?
Oh.
Oh, how nice. Oh, this is... Oh, y'all are so lovely.
I only have 1 year left.
Oh my gosh. This has been going on all last semester. You do plump. You start to go down. Yeah. Yeah. Okay. Just hurting my heart a little bit. Okay. So growth hormone secretion disorders. All right. We can have two different problems here. So if we have a deficiency of growth hormone, we get pituitary dwarfism. You're not squirting out enough growth hormone, so you're not able to grow taller. You have lower blood sugar levels. You have lower IGF-1 levels. You stay short your whole life. This can be a problem in the pituitary or a problem upstream in the hypothalamus. If you're squirting out too much growth hormone, then you have pituitary gigantism. This is deadly. This is too much growth hormone. It is. There are a lot of deadly things. Too much growth hormone, if it's not regulated, you get visceral hypertrophy, so all of your internal organs get so big, your body can't sustain them. Your cardiovascular system is overwhelmed, and you die. So it has to be treated. Okay, acromegaly is excessive growth hormone production in adults. This is displayed primarily by facial bones and hands and feet bones getting very, very big. So you have very large hands. Very large hands. A big face. Increases the release of glucose from your liver because remember, growth hormone stimulates the release of glucose from the liver by mobilizing glycogen. Your internal organs will increase in size, and you typically end up with this when you lose hormonal sensitivity. So you either downregulate too many receptors that are receiving the negative feedback signals to stop growth hormone release, or you have damage to the tissues expressing those receptors. Okay, so we'll stop with that and move on to your sheets. Thyroid will be on Monday.